Well, thank you. Thank you for coming today. We are going to, as David Bushman was given the first introduction, during the day we have to thank a lot of people. David is the primary organizer of this event. He mentioned that this test, as you see, there is no name. He said it's just we are going to collect all the goal here is that the majority of the group, we are going to have a summary of the answers, and then for continuing medical education, continuing nurse education, all form of education, we need to have pre-test, post-test for us to, this is an evaluation of ourselves, how well we perform, in this review course. As an introduction to this course, we have we are from the Norton Infectious Diseases Institute, part of Norton Healthcare. All the presenters are members of Norton Healthcare, and the goal of this clinical review course, you already have the objective. The primary objective is to give you an overview of clinical research. But we think that having an overview of clinical research is important for two reasons. One is that some of you may be interested in the topic of clinical research, and this may be a refresher, or you may be better prepared to perform clinical research. At the same time, we think that everything that when we advance in medicine, every new advance is through the development of clinical studies, to clinical research. This clinical research we are going to publish. I think that a better understanding of the process of clinical research also is going to help us to better read the literature, and better for us to then, even though I may not be doing clinical research, I need to know the basics to be able to analyze the literature. And whatever I'm doing in healthcare is going to be important. When I read, I say, oh yes, I remember this is the methodology. Are they doing a good job or not a good job in this manuscript? We were discussing with some of you this morning that with the problem that we have now with the COVID pandemic, for the last 2 years, everybody in healthcare have been doing research in COVID. And we were desperate for new information because we have our patients. Remember, this is March 2020. All here we're in Louisville, Kentucky, for the people that are in Zoom in other countries, because in Asia the problem was in January, in Europe was in February, in the United States it was in March, but it's March 2020. We have patients in our hospitals. Patients are dying of COVID pneumonia, and we don't know what is the management because we didn't have any information. And it was due to clinical research that we started learning how to manage these patients. And whatever we do now is based on clinical research. Then COVID has brought to the front the importance of clinical research, because if we are going to advance medicine, it's through clinical research. But also COVID brought to the front the importance of reading the literature properly, because since everybody was doing research in COVID, there were so many publications that they were the full spectrum, the very good and the very bad. And we were in the middle trying to do the best for our patients. And then the question is, do I need to use this? Do I need to apply this procedure? Do I need to do this diagnostic test? Well, you can find everything in the literature. Yes, you are supposed to use hydroxychloroquine. No, you're not supposed to use hydroxychloroquine. Yes, you are supposed to use azithromycin. No, you're not supposed to use azithromycin. This was at the beginning. And we were reading the literature to try to do the best for our patients. Then this is another objective of having a refreshed course of clinical research. When I look at the literature, I know what is going to be good evidence and what is going to be bad evidence. Then this is as a form of introduction of what we're going to be doing during the day today. We want to, this course is done in partnership. I mean, who was working with us putting together these presentations? Members of the Norton Infectious Diseases Institute. We have members of Norton Healthcare. We discussed this. The International Respiratory Infections Society is a society that the goal of the society is to improve or to facilitate research in the area of respiratory infections that we work with the society. The Kentucky Infection Prevention Best Practice, Best Outcome, there is another group trying to improve infection control and infection prevention throughout Kentucky. We are working with Haymarket, that is a national CME company, and this course is going to be hosted in what is called myCME, and then people are going to be able to look at this course in the future. And we have one sponsor, one company is Gentex, that they have a booth there outside. And we want to be, we are very grateful to Gentex that they saw the importance to support this idea of helping us to better understand clinical research. We are going to, exactly at 8:30, we are going to start with the first presentation. We are pleased that we initially were planning to be at Children's Hospital. We have an auditorium there for 30 participants. We have much more participants that wanted to attend this in person. We have participants via Zoom. Then we try to maintain the time of each presentation. I ask all the speakers to be sure that we can maintain exactly the schedule. And the presentations are we have a very tight time. Then the idea is that if you can, as the presentation goes, you have the possibility to write questions, and please write the questions. The goal is that at the end of the day, we are going to have a roundtable discussion with all the presenters, and I'm going to moderate the roundtable discussion. At the end of the day, you are going to have the opportunity to present questions, but you are writing the questions during the day. This will allow me to characterize the question and ask the questions to all the presenters. Then at the end of the day, we're going to have 1 hour of just, if there is something related to clinical research that we didn't address, but you have a particular question, just send us the question. For the group, for the last group that is connected via Zoom, if you have any question, please send that in the chat, and we are going to be collecting the questions. Then the idea is that by the end of the day, we want to be sure that everything that we wanted to ask or discuss regarding clinical research can be addressed. David already did the consideration that where is going to be the food, where are the restrooms. Any other things that we need to mention before we start, David? Any other general? Okay, then we are pleased that we have 12 countries represented in this clinical research course, and then we are excited for us to be able to present, but we're also excited for the questions that we're going to be able to send. Then this is the first year that we do this in our with Norton Healthcare. This is going to be an annual course that we are going to have at Norton. And David mentioned that if for each section is going to be questions, please be sure that it's not mandatory for you to answer the question, but be sure that you can answer the question for us to have a good idea of what is the basic knowledge and what do we do after each one of these presentations. Then I'm going to be introducing the speakers during my first presentation. The structure of the course is that I'm going to have one initial presentation that's going to be a summary of whatever is going to happen during the day. Then the initial presentation is an overview that then you get in 40 minutes, an overview of what we're going to be discussing during the full day. And then, as I mentioned, each one of these presentations is going to be taped, It's going to be on myCME for if you want to review this with more detail. It's going to be available in myCME. Then with this, we are going to move. Any other considerations for this introduction? I'm looking at the speakers in the front row. Okay. Then, as I mentioned, in the first presentation, I'm going to give you an overview of the topic of clinical research. This is a 40-minute presentation that I try to cover everything that we're going to discuss today. And to give this overview, I'm going to discuss some clinical research definitions, the planning the study, performing the study, analyzing the data, and publishing the study results. This is the structure that I'm going to follow today. If we look at clinical research definitions, essentially, study for research. Research, the definition of research probably have two components. Research is the generation of new knowledge. Then we need to generate new knowledge, new information, and the dissemination of this new knowledge. Then when we were to look at research, we hear creation and dissemination of new knowledge. Then in biomedical research, sometimes we talk about basic research, the research that we do in the laboratory, and clinical research, the research that we do working with patients or with humans, human research. And the primary goal of clinical research, as we mentioned, is the idea is that I'm going to generate new knowledge with the primary goals to improve patient care. Then if we are going to do something better in healthcare tomorrow, it's going to be primarily to clinical research. Then you are in healthcare, you try to, you have a question, and you try to say, okay, what should I do in this particular situation? How do I approach this patient? What diagnostic tests or what should I do? Then you look at the evidence. You go to the literature and look at the evidence. When we look at the evidence, you look at the new information. Sometimes you see in the literature the pyramid of evidence. The idea is that some of the information looks very good and some information is weak. And at the bottom is laboratory and animal studies. Now we say, well, I don't manage patients based on what happened with a rabbit or what happened in the cells in the laboratory. Well, yes, because we look for better evidence. But remember, I go back to the idea that 2 years ago with COVID, in March, the only thing that we knew is that SARS-CoV-2 inside of a cell was inhibited by hydroxychloroquine. And because of this inhibition, we jumped from the cells and we start using a drug in humans because we didn't have any other better evidence. Of course, if we have better evidence, then it's simple. I move up. But then all this area of evidence, if we want to move up in this pyramid, is through clinical research. And anyone in healthcare today, whatever you're learning, you are in the nursing school, medical school, whatever school you are going, whatever you're learning today is something different or 10 years ago is because of clinical research. Then we need to emphasize that this is a critical aspect of healthcare. Now, usually talking about definitions, we consider that you are interested in an area of clinical research, and usually an area maybe a health problem that we call the target population. You are working on diabetes or cancer. In our case, we're working on respiratory infections. Then this is your target population. And let's say that you are working on COVID. Then everybody with COVID all over the globe, this is your target population. When we're doing research, we're always going to discuss that is the study population. Then these are the patients that you're going to have in the study. We are interested in COVID, and you are interested in COVID pneumonia, and you are here in Louisville, and in your hospital, you're going to follow the 200 patients with COVID pneumonia. This is the study population. And from these 200 patients with COVID pneumonia, you're going to see if this medication works or not, or this diagnostic test is better to diagnose SARS-CoV-2, and you're going to generate new knowledge. But the idea is that this new knowledge that you generate is not just for the 200 patients, it's for everybody with pneumonia, with COVID pneumonia in the universe. Then the new knowledge needs to be generalized to the full population. To do this, we plan the study, we do the study, we analyze the data, and we publish the results. And this is going from idea to publication. Excuse me, could you let me know at what time I was supposed to finish? I was complaining about timing, but I forgot to check at what time is the end of my presentation. 9:20. Okay, thank you. Because I can't keep talking the full day. I have to be at my 9:20. Okay. Then going from idea to publication. And then most of you are familiar with circles of quality improvement. No, plan, do, study, act. There are different ways to look at quality improvement. Well, we have our circle of improvement in research. You plan the study, you do the study, you analyze, you publish, and then you continue with the circle. You plan the new study. And at every time you have a research team, you go to a research team, people are going to be doing a different area. We are planning this study, we are doing this other study, I'm analyzing this data, I'm working on this manuscript. You're always working in different areas of this continuum of clinical research. Then we are going to move then to planning the study. And I'm planning the study usually then start with the research question. I mean, what is that? If I'm interested in COVID pneumonia, okay, what's going to be my questions for the patients with COVID pneumonia? Do we need to use antibiotics? What is, if I'm still trying to work with something, remember that there was the question of steroids, that steroids work for COVID pneumonia. And at the beginning, if you look at the pyramid, there was all expert opinion at the bottom. And people say, well, steroids don't work for influenza pneumonia, are not supposed to work for SARS-CoV-2. Don't use steroids. Another expert were saying, but COVID pneumonia has a cytokine storm, steroids may be good, let's use steroids. We didn't have any clinical studies. We're all expert opinion until we have the clinical studies. But let's say that if your question was, can I use steroids for COVID pneumonia? Let's go to a question. Now we have this controversy in regular pneumonia for us. Patients admitted to the hospital with pneumonia. Do I need to use steroids? Then you come out with your research question. And to address the issue of the research question, we are going to have a full presentation that Ruth Carrico is going to discuss with us. What are the characteristics of the good research question? Because this is the beginning of the process. Once you have your research question, you're going to start working on the study protocol. And the study protocol has different components. And most of the time in research, we always work from an outline. We are giving you here the outline of the full process of clinical research. Then in the protocol, it's going to be a rationale, objectives, hypothesis, site design, what subjects you're going to study, what variables you're going to collect, how many patients do I need, what is going to be my statistical plan, and then you start developing your protocol. Sometimes you read in the literature that your research question, your hypothesis, you have your white paper or your letter of intent or this is just 2 pages. But the idea is always that the ideas needs to go into a paper. Because when you start writing, you start clarifying your thought process. Then you say to a person, someone can, I have this idea, okay, put it on paper, and this is my idea, and this is my hypothesis, and this is how many pages are going to, and you start with 1 page, 2 pages, but then it's going to end with the 20 pages that's going to be the study protocol. When we look at the study design, what is going to be my study design? We usually discuss that there are observational studies, that these studies are studies in which I'm not going to do any intervention to the patient, as we're going to observe what happened with the patient. We are going to have interventional studies that, yes, I'm doing something to the patient, trying to change the outcome. And then there are multiplicity of studies designed that can be observational studies, will be interventional studies. Then Ashley Wild, another member of our group, is going to discuss. We are going to have a full presentation of observational studies. And I have to tell you that this is what moved medicine. This is what moved healthcare, observational studies. Even though the primary things that you always read is the interventional study, the prospective double-blind randomized clinical trial. And for this, Stephen Formanek, another member of the group, is going to discuss interventional studies. But interventional studies are usually more, they are at the top of the pyramid, but you need a lot of support, a lot of money, these are more difficult to do. Most of the time, what we are going to be doing when we start in clinical research is going to be observational studies. But you're going to have one presentation observational studies, one presentation on interventional studies. Another way to classify the study design is where I'm doing the study. I mean, the study is generated by whom? Then you can say, well, I have studies that are done in a university setting, large medical centers. This is the traditional way to do clinical research. But now it's also important that sometimes we do studies in the community. This is what is called community-based research. Or studies in other countries based on the needs of the other countries. Or in the other country, they have a need. The problem in the other country is malaria. Okay, I'm going to go to the other country to work on malaria. It's not that. Then these want to be, these are interventional studies. And then again, Ruth Carrico is going to give us an overview of community-based studies and interventional studies. This is another way of my study design. In this case, the design is not based on observational intervention. It's based on where I'm going to do the study. Another possibility to classify the studies is that, for instance, you say, well, I have the study patient, and if I'm going to do an intervention, a new drug, I'm going to draw blood and do these new laboratory tests, I'm going to check the cytokine levels, then you're going to have a lot of study procedures and results that are based on the study. I'm doing the study, I'm getting all this new information. But at the same time, there's a possibility to do a study that based on the practical, based on what we do every day with the patient, and just collect the information of actual medical practice. Then the study data is based on whatever happened to the patient every day. And this is what is called real-world data. And this is what is called real-world evidence. And then the idea is that these are studies that I use exactly the way that I treat the patients every day. This is a new area of clinical research that is taken a lot. And now that the FDA even approved the consideration of studies using real-world data, real-world evidence, this is growing. But here the idea is that, for instance, we are in our case, Norton Healthcare and Norton use an electronic medical record. It's called EPIC. Then you look at the electronic medical record. This all the data of all the patients. You just look at the database with electronic medical records. You're doing studies using real-world data. Then this is another way to classify research based on if you're going to be using data considering actual medical practice. Then this is a little bit of the different study designs that you are going to be thinking, okay, based on my question, what is going to be the best study design that I'm going to use? Then we discuss that it's going to be the subjects. I mean, what type of patients are going to need for my study? I'm going to have inclusion criteria, exclusion criteria. What variables? Variables, everything that I'm going to collect. Then we have the predictor variable. In this case, okay, I'm going to use steroids. The outcome variable. If I'm going to try to use steroids in pneumonia, the outcome may be mortality in pneumonia. The confounded variables. The variables that are associated with my predictor and my outcome. What is going to be the sample size? Is this a prospective study? How many patients are going to need to enroll in my study? Then all these concepts, we have a particular presentation on study subjects, study variables, sample size, that Thomas Chandler is going to present, another member of our group, that is going to give you an overview of this topic. And still, we are still working on finishing my study protocol. Okay, I'm still planning the study. After I finish the study protocol, there's a lot to write before you can start the study. And these are all other documents that needs to happen. Then you have the study protocol. I'm going to do the data collection form. I'm going to do the manual of operations. I mean, how is that I'm going to do everything that I want to do in the study step by step. I'm going to develop the study database. I'm going to have a data dictionary to be sure that whatever is in the database is clearly delineated. I'm going to have my regulatory documents, because remember that every time that you have to do research, you need to ask for permission to do research. And this permission usually happens from the IRB and other areas. Then you are going to do your study budget, because remember that every time that you spend time doing anything, there's cost, and every study has to have a budget. And these are all different documents. And then we are going to have a particular presentation on data collection and database. Now, I have to tell you that Bill Mattingly, another member of our group, developed COVID yesterday, and now he almost lost his voice. Then last night we mentioned to Stephen Formanek that he's going to give another presentation. Stephen is here. Then when Bill developed COVID, we say, okay, Stephen, you're next online. Then it's going to be Stephen Formanek discussing a data collection form and a study database. Now you say, well, what the data collection form has to do with study database? Well, working year after year, we figured out that the person that developed the database, the statistician, the biostatistician is going to develop the database, needs to be involved in doing the data collection form. Because when you start doing the data collection form, automatically you are developing the database. Your data collection form is the outside face of your database. This is why we say that we need to have these two presentations together. Also bringing the point that clinical research, as so many other things in healthcare, is a team work. Don't try to do clinical research on your own, because you are definitely going to fail. Okay, then now you say, well, I don't have a team. How do I do? Well, you need to, either you develop a team or you join a team. But this would be the saying that, oh, I'm interested in doing medicine, I'm just going to do medicine. No, you need to have a healthcare system to do medicine. You cannot just do medicine, be a family doctor, go to a town and open it. No, this doesn't exist anymore. You cannot do clinical research on your own. Then either you develop your own team, that's going to be difficult. You have to join a team to do clinical research. Then Stephen is going to give this presentation. Ethical and regulatory considerations. We mentioned we need to understand the ethics and the regulatories. In this regard, Ashley Weil is going to give us an overview, ethical and regulatory considerations. Now, all these documents that we discuss, sometimes you say, well, but I'm doing clinical research. It's going to be a small study. I don't need to have all these documents. But the reality is that all these documents is what is called the study proposal, or sometimes this is called the grant proposal. Then if you are submitted because you want to get funding, this funding may be $20,000 from the research office at the university or from the foundation to $2 million from the National Institute of Health. Of course, but even for the $20,000, even if you go to a small company, you will need to show all these documents. You need to have a protocol, you need to have a data collection form, you need to have a database, you need to have the regulatory documents, you need to have a budget, because even if you ask for $20,000, they're going to say, okay, what do you want to use the $20,000 for? Then all these documents are going to be important for your grant proposal. Now, sometimes all these documents may be coming from industry, and sometimes then essentially what we have is a drug company is trying to test a new drug for cardiovascular disease. Well, the company put together the question, the study protocol, they suggest a budget, they have the database, and they are looking for us to do the study, to do the next phase of the study. This sometimes happens with the phase 3 clinical studies coming from drug companies. But then this will be an industry initiated study. But then we are discussing here, if you're going to be the investigator initiated study, this is going to be the grant proposal. This is what you're going to submit to the Bill Gates Foundation to see if you can do this type of research in this particular topic. The study budget and the grant proposal. This is then Jeff Weiss is going to give us also from Norton Healthcare, from the Office of Research, is going to give us an overview. Now, as you can see, each one of these topics, you can have 3-4 hours discussing each one of these topics. Now, we have only 8 hours and everything is going to be 20 minutes, okay? But we want to give you the overview, then you are going to have something more clear in your mind to go deep into each one of these topics. Then at this point, we past lunch, we're in the middle of the afternoon, and we just finished the planning. Okay, these all presentations is how to plan the study. Now, and planning is the easy part. Okay, planning is you get with your friends, have coffee, type, this is planning. Okay, there's not too much. Then the part that kills most of the studies, the doing, okay, performing the study. Because, talk and writing is cheap. The question is, who's going to do it? And how well you're going to do it. Then, and this is why you need to have a team, because doing a study is very difficult. Then we screen for patients, we have to enroll the patients, we need to have the study procedure, going to draw blood, get urine, get a chest X-ray, whatever is part of the study. We are going to follow up the patients in the hospital or in the clinic. We are going to do data collection. We are going to enter the data into a database. We need to do data quality. We are going to go for all these queries. And finally, one day, we want to say, okay, we are done with the study. We log the database. We finish. Now, this is the area where most of the mistakes happen. Because you have 20 people doing something, mistakes are going to happen always. Then the question is never an issue if there are mistakes in clinical research. The question is, how many mistakes do we have? And during data quality, have we been able to correct most of the mistakes? Then if the study ended, we log the database. We have some mistakes, but we want to, something that we can consider that is acceptable. Then we log the database. Then this one presentation for doing this study. And again, Ruth is in charge of giving everything that you have to know to do the study. Patient enrollment, data collection, data quality. But the reality is that there's no way to give a presentation doing the study. What is the only way to learn how to do something? You have to do it. How you teach someone to ride a bicycle? There's no way to teach. You have to get into a bicycle and push you and here you go. Then the way to learn this is by doing clinical research. But we are going to have the structure, but this is how we do the study, the elements. Okay. We discussed that you mentioned 9:20. Thank you. Yes, yeah, no. This is we discussed that then at this moment we plan the study, we perform the study, we log the database, we have the study results. By the time that we log the database, if you have a research question until you log the database, an average is 1.5 years, 2 years. Then you may say, well, I'm doing a residency program of 2 years. I don't have 2 years to do a study. And then what do I do? Well, the question is, can I do clinical research without doing the study. That is the most difficult part. And I may say that anyone that start, this is why I said, you start with a team, you're going to be doing the research. But what are the alternatives? The alternative is the ancillary study. The alternative is someone else is already doing the study. This group is doing this large study on steroids and community-acquired pneumonia. And they are collecting 300 patients with pneumonia, 150 is going to get steroids, 150 are going to get steroids, and you know what they are doing because you are part of the group or you know what they're doing. Then you say, okay, you're doing this, you're collecting these 300 patients with pneumonia. I'm interested to know if procalcitonin level, there's a correlation of positive blood culture with procalcitonin. Can I add to your data collection form level of procalcitonin and result of blood cultures? Then someone else is doing the study. You just ask a question to an study that's already going, and you have your study attached to another study. This is an ancillary study. Then you know that someone else is doing a study. You say, oh, I have another question for you. Even a more economic way is that someone already did the study, and they finished the study with the 300 patients with pneumonia, and they randomized to steroids and non-steroids, and then analyzing the data. And you say that, have you collected procalcitonin? Well, procalcitonin is part of the standard of care in the hospital. Blood culture is part of the standard of care in the hospital. You go there and say, I have this question, and I'm going to ask the database the question. Can I use your database to look at what happened with my question? Then essentially, you're already doing the study. The study was done by someone else. You ask the database the question. Another consideration is that you know what is in your medical record because you are in a particular hospital. You know what is in the medical record. You know that every patient with pneumonia has blood cultures. And your question is, what is the incidence of positive blood culture? How common is multi-resistant Pseudomonas in positive blood culture? You go and ask the database that is the electronic medical record. You see that this will be real-world evidence that you're looking. Then it's going to be a lot of questions that you ask that you don't need to do the study. You ask a database. Then we finish with the study. Then we log the database, and then we are going to start analyzing the data. And from the very beginning, you have your predictor variable, your outcome variable, and in clinical research, everything is, do we have an association that this causes that this is associated with that, and is this association is real? This is the issue for us in clinical research. Is this cause effect? And then in clinical research, let's say that I finished this study. I decided to do, let's make it simple. I know that some patients in the hospital get steroids for whatever reason, other patients don't get steroids. Then for my question of steroids or pneumonia, I'm going to go to a medical record and collect 300 patients with pneumonia and figure out who has steroids, who didn't get steroids, and see if patients will receive a steroid because they have COPD and we just give a steroid for something else. And figure out, you get steroids, you decrease mortality or not. Now, I get the 300 patients, I do the analysis, and in my 300 patients, using steroids decrease mortality. Then I have an answer. Steroids decrease mortality. This is the answer in the study. This is the answer in the 300 patients. Now remember that the answer decrease mortality in everybody with pneumonia in the world. I'm not interested to see if a steroid decrease mortality in my patients. I want to generate new knowledge for everybody with pneumonia all over the world. Then steroids decrease mortality in my 300 patients. These results could be true or false. Will be true if a steroid decrease mortality in everybody all over the world. But it will be false if steroids don't decrease mortality. And every time that you finish a study and you have a result, you don't know if your result is true or not. Now, why your results may be false? There are three factors that explain why your result may be false. Maybe confounding, maybe bias, or maybe chance. Then every time that you analyze your study or the study that you're reading in the literature, tomorrow you're reading an article, and the conclusion of the 300 patients is that steroid decreased mortality in pneumonia, there's still controversy. We don't know if we need to use it or not. We know that we need to use it in COVID because in COVID decreased mortality. But in the rest of pneumonia, we don't know. But I finished my study and said, the steroid decreased mortality. Well, the study results may be true, maybe due to confounding, due to bias, or due to chance. Now, we are in the analysis phase of the data. I finished my 300 patients. I reviewed these 300 patients. I found 100 with steroids for other reasons, 200 without steroids. I give it to Thomas, to Stephen. They do the magic. So I give it to my statistician. They do the magic. They do the hypothesis testing, the p-value, and they give me back a p-value. With hypothesis testing, I want to discuss this more in detail. I'm just giving you an overview. Statistical tests or mathematics, I would say here, cannot be used to say that a hypothesis is true. Okay, the statistical test can be used to tell that a hypothesis is most likely false. Now, this for us is important because sometimes we complain that we don't understand the statisticians, what they talk. But the problem, they don't understand us, what we talk. Then we need to have a better understanding because we have different languages. Then how do we translate our thought process to the way that a statistician thinks? Well, this is the rejection rule. The statistician is going to do mathematical modeling to see if the hypothesis can be rejected. Now, what is the hypothesis of my study? I'm looking at if steroids decrease mortality in pneumonia. My hypothesis, what I want to find is that patients with steroids are going to have decreased mortality compared with patients without steroids. This is my hypothesis. Steroids decrease mortality. Now, since my hypothesis, I don't want to give my hypothesis to a statistician because the only thing that they can do with a hypothesis is destroy the hypothesis. Then what do? I make up a different hypothesis. Then I'm going to do the null hypothesis. Okay, then the null hypothesis is that steroids has nothing to do with mortality. Steroids are not associated with mortality. Then this should be the null hypothesis. This is you see the hypothesis 0 or again, we ask our statisticians that we have a rule that in the clinical research course, we cannot use formulas. Okay, they are supposed to give us statistical talks without using formulas. So we are there. Then the null hypothesis is the hypothesis that's going to be rejected. This is the hypothesis that the statistician is going to work. The alternative hypothesis, when you read an article, the null hypothesis, the hypothesis of no value. In this case, steroids are not associated with mortality. The alternative hypothesis is that steroids are associated with mortality. This is a correlation of steroids and mortality. My study hypothesis is that steroids decrease mortality. This is what I want to find. The alternative hypothesis is that steroids may decrease mortality or steroids may increase mortality. Steroids are associated with mortality. I don't know which way. Because sometimes we are studying one drug and just happen to be that the drug is worse than no drug. Then it will be the alternative hypothesis. Then the null hypothesis presumes no association between predictor and the outcomes. Steroids and mortality are not associated. The null hypothesis is rejected when the p-value is less than 0.05, that this is 5%. Then the alternative hypothesis or my study hypothesis, I cannot be tested directly. I accept the alternative hypothesis when the statistician said we can reject the null hypothesis. Then, and we want to discuss this during the day. Then the statistician said, your data, the p-value is less than, equal or less than 0.05. Let's say your p-value is 0.001. We are all happy because it's a very low p-value. And then my study results, again, we mentioned, I want my study result to be the truth. I want my study result to have minimal confounding, minimal bias, minimal chance, and be the truth. Then what will be the answer to this question? My data says the steroid decrease mortality. This is a very good possibility if my study is true, meaning in the universe, steroids are going to decrease mortality in all patients hospitalized with pneumonia in every hospital all over the world. Or it's a low probability of confounding, or a low probability of bias, or a low probability of chance, or all of the above. That will be, I'm really close to the truth. Then this is just a mental exercise for you to get the answer mentally. And then we're going to emphasize this during the day, that the p-value, the answer for this is D. Okay, the p-value is a low probability that the result is due to chance. When I'm doing this course, at the university over the years, and I will have to have a paper, and everybody will write the answer. And the average is 50% get it wrong. And these are 50% for medical students, residents, everybody have been in medicine for years. And still 50%, and we all reading articles every day, and still 50% doesn't know how to interpret the p-value. Then it's just the possibility of chance. Then we are going to emphasize this over and over. It's a mistake to believe a research study just because the p-value is low. You have, again, 4 possibilities: truth, confounding, bias, and the p-value is going to discuss only the consideration for chance. Okay. We do the statistical analysis, and then we are going to do also the clinical analysis. Now, we mentioned that with the statistical analysis, and there's a lot of tests that you can do for statistical analysis. Now, I mentioned here probably the most common test that we do in clinical research. Now, you have books of statistical tests, but you see the hypothesis testing and p-value is going to give you the consideration, which, okay, what is the possibility of chance coming in your study? Now, it's also interesting that we discussed the p-value is 0.05, meaning 5% of chance. We accept that my results, if I repeat the study, the possibility that I'm going to find my results by chance is 5% or less. Now, from working in healthcare or just in life, if I say you have a 5% chance that this is a mistake, or you have a 6% chance that this is a mistake, you find any difference between 5% and 6%? There's no difference. But from the statistical point of view, 5% is the magic number. Now, this magic number is, these are the statisticians come up with this number. It's not us. Okay, then what I try to say is that if the p-value is 0.0, if the p-value is 6%, it's not to me, it's not a big deal as a clinician compared with 5%. But this magic number of 5% is just magic generated by, not by us, generated by the statisticians. But we accept the 5%. Then Stephen is going to try to convince us that the statistical significance hypothesis testing at these 5%, these 80%, these magic numbers is the thing that we are supposed to use in clinical research. Then Stephen is going to give us a presentation on statistical significance and hypothesis testing. Now, we mentioned that another factor that is a problem when we analyze is confounding. Everybody knows the story or confounding of coffee drinking and lung cancer. Okay, yes, coffee drinking. Every time that you read a statistic, this is the example. You drink coffee, you get lung cancer. There's a strong association. The more coffee you drink, the more lung cancer. There's a strong association of coffee drinking. But we know that the story, and this is the problem of confounding. Then sometimes you say, is there an association between coffee drinking and lung cancer? The answer is yes, there is an association. The problem is that it's not cause-effect. Okay, there is an association. Then you have all these, and this is a real association. Then you have all these jokes that you read in clinical research tests. I was telling Ruth the other day an association in a real study on people looking at if driving using a cell phone is associated with you crash your car. And then someone look at, okay, what time people crash their car, and they're looking at the telephone records, and yes, you were on the phone and you have to have an accident versus you were not on the phone, you have an accident. Then there was a strong association with very significant p-value. But the problem was that you cannot find exactly the time. The problem was that every time that you crash, immediately you get your phone to call the police or call for help. Then, yeah, there was an association, but it was not cause-effect. It was There's an association between coffee drinking and lung cancer, but it's confounding because it's smoking and lung cancer. But if you smoke, you drink coffee, and the more coffee you drink, more you smoke, then there is association, and then there is this confounding. Then controlling for confounding, that is critical for us in clinical research. We can control for confounding when we design the study, in the planning and in the analysis. And then Thomas Chandler is going to give us a presentation of controlling for confounding. And finally, there is an association between steroids and decreased mortality, or really there is an association between this antibiotic and better outcome. There is an association because this vaccine decrease COVID. Then how strong is this association? How many patients do I need to treat to get one benefit? Because this is going to be all important considerations when we analyze the data. Then these are three elements that I want to discuss from a statistical analysis. Now, after the statistical analysis, I'm going to perform the clinical analysis. We're going to, because this is going to give us the clinical significance. When people read the question, so what? Who cares? Okay, to answer these two questions is how clinical are my clinical relevance of my results. Now, we mentioned multiple times, we're going to mention multiple times, statistical significance is not clinical significance. These are two different things. This is a problem in clinical research as so many things in medicine. First, we need to agree on terminology. And the problem is that the statistical terminology, the statistical language is different from the clinical language. What the statistics call statistical significance is not what we call clinical significance. These are two different things. Then you can have a study that is clinically significant, statistically significant, but not clinical significant. Then a very low p-value doesn't mean that I need to be using this with my patients. I need to figure out the clinical significance. And then Dr. Joseph Flynn is going to give us an overview. Again, each one of these topics is for a long discussion, but we have only 20 minutes. Then Dr. Joseph Flynn is going to give us an overview. How do we look at the data, and how do we know that this data is clinical significant? Then with this, we finish with the analysis, and now we have the study conclusions. And then we are going to publish the studies. And they publish, as everything that we do, there is a structure, there is the introduction, methods, result, discussion, and this is going to be the way. And if you notice, if you've done the study, the manuscript is almost all done, because the introduction is what you have in your protocol. your research question, your objective. The methods, you already have it in your protocol, what you're going to do. The results is all your analysis. You already have your results. And part of the discussion is your clinical interpretation of the data, the clinical analysis. And then you're going to publish, but even for this, you need to have a team. It's very difficult to write one manuscript on your own, because you write, you send it to your group. It's the group, you have version 1, send it to your group, get feedback. Version 2, send it to your group, get feedback. At least in our group, I don't know, we may be slow, but before we send in any manuscript, usually it's what version? Version 15 to 20 for every manuscript that we're going to submit. Okay. Then it's write, rewrite. And as we always mention, you write. Someone from the group said, this is not clear. It's not the problem of the person. That, don't try to explain. No, what happened? No. If something is not clear, it's your problem. You have to make it clear. Okay, don't try to explain it to the person that is reading why he doesn't get it. No, if he doesn't get it, it's your problem. Because everything has to be very clear. Then you submit all the manuscripts. Your baby is beautiful, okay? Because this is always the case. My baby is so beautiful. Then, see, my baby is beautiful. Where am I going to submit my manuscript? To New England Journal of Medicine, because it needs to be in the New England. And then you figure out that your baby is not as beautiful as you thought, because it gets rejected. Well, let me send it to the Annals of Internal Medicine, rejected. Oh, my goodness. Let me send it. Finally, I want my baby to be published in any place. Okay. But you resume until your reality check and say, well, you just want this to be published. And then finally, you get a publication. Okay. And remember the first thing that we mentioned, research has 2 components. You generate new data, you generate new information, but you need to publish. The worst thing that you can do ever is to finish a study, have some information, and because something happens, you just put it there and die. Essentially, you have not done research. You have not done anything. On the other hand, you've done your research, you finish, you publish. Then, in healthcare, if you are a practitioner, because you're a nurse practitioner, doctor, resident, you treat one patient. You help one patient. If you teach 5 residents, 10 residents, you know that these people are going to help, are going to treat 10 patients, you're helping 10 patients. And teaching is a way to helping more patients. With clinical research, you move the needle, you treat everybody all over the world. Then you are moving the needle. If someone in the school of nursing or the School of Medicine is going to learn something new in 5 years, it's going to be because we move the needle with clinical research. Then it's a way to help everybody in the universe. But you need to publish your research. Then this is so important that disseminating study findings, scientific writing. Then again, Jeff is going to give us an overview of this. And then essentially, at this point, then we finish with the publication. And we mentioned in every clinical research team, you're going to see that we are planning studies, we are trying to develop ideas, you're planning protocols, you are always doing some studies, you are going to be doing data analysis because there are always some data analysis, and you're always working on some manuscripts. Because in healthcare, our approach, as so many people approach, is everything that we do in healthcare, that there is some teaching. It is something that someone else needs to know. The only way from someone else in a different hospital to learn what you improve in your institution, you need to publish. And everything that we do, there has something, you have something to say, that's going to improve healthcare needs to be published. It's something to say you need to teach, you need to have students, and you need to publish to be sure that the person in China and in Chile is able to apply what you learn in Louisville, Kentucky. And this is the idea of the publication. And then we are going to finish the day. At the end of the day, all the instructors are going to sit here. We're going to have a roundtable discussion, and all the questions that you're going to have during the day, and every question that you want to ask directly, we're going to ask to all the participants. Then this is what is going to happen during the day today. This is the overview of clinical research. As we mentioned, then I give Ruth 2 minutes, 2 extra minutes. And then the next presentation, then Ruth Carrico is going to give us the concept of what is a good clinical research question. Ruth?